Chapter 9. Testable Elements of the Hypothesis. One initial approach would be to seek evidence for or against some fundamental aspect of the working hypothesis, test for the existence or lack thereof of the proposed entanglement cells or EC that establish and maintain life via the QE connection in complex hosts. Termination of the host's ECs and no other cells should result in the termination of the subject. Premise: can death be induced without damage? Can an otherwise healthy living subject be terminated with empirically no physical damage contributable to subject termination? Barring any limitations of technical proficiency or of equipment in analyzing and identifying the root cause of subject death. Axiom: there exists some absolute minimum number of cells that may be terminated in any complex organism, whereby such cells may be scientifically established to be the root and only cause of death of the subject organism, with no pre-mortem adverse effects to other cells in the subject. Cells that meet these criteria are candidates for the theorized entanglement cells, and the collection has a high probability of including some or all of the subject's proposed entanglement cells. Practical test: perform controlled experiments using approved subjects, that is, fruit flies, to terminate the minimal number of cells per specimen to conclusively induce death of the test subject. Carefully repeat and document the number and location of target cells per subject for each scientifically substantiated successful sample. Repeatability per species is mandatory, as the specifics may vary from species to species or subject to subject. In qualifying samples, the cells that are the root cause of death must be gradually minimized and physically isolated. Cellular damage must be limited to only the target cells for a duration beginning at the time of the target cell's death up to and including the time of confirmed subject death. In other words, for a successful trial, no cells in the subject other than the target cells may be adversely physically affected pre-mortem. Neither immediate acceptance nor rejection of a naturally invasive, scientifically plausible idea will serve to either validate or invalidate such ideas. It is only by proper testing, and perhaps with a few unexpected discoveries along the way, that its eventual accuracy and relevance will be determined. Interested parties should first only seek to comprehend the details of what is being suggested without an emotional predisposition one way or the other. It is always persons affected emotionally by the implications of what is being proposed that often presents early, emphatic opinions on a new idea. This is perhaps as it will be. However, calmer heads and nimble minds are always present and should consider and ask probing questions as they see fit. The proposed test is one approach to identify and locate the subject's hypothesized entanglement cells via a systematic decremental process of targeted termination of candidate cells within the test subject, that is, fruit flies, nematodes, etc., and thereby to finally terminate a healthy subject by disrupting only the subject's entanglement cells, while inflicting no damage to the host's non-EC cells, ergo death without damage. Today, all death known to modern science is eventually forensically caused by cellular damage to structures singularly or collectively vital to the host form. Such damage can invariably be determined to sufficiently disrupt conditions required for proper host function, thereby resulting in the termination of the emerged individual, death. The line hypothesis describes death as the disentanglement between one's entanglement cells, or EC, with metamatter. This results in the loss of the individual's position of view, or POV. Today we see only the physical symptoms of the damage to the host, and we quite adequately associate these conditions with the termination of the individual. This is fine for all that we currently do. However, this is not the complete description of life in this universe. If indeed it is the sole function of the host's EC to maintain life in the emerged individual, and if it falls upon all other cells of the host only to maintain the environmental, internal, and/or external conditions for the individual's continued function, then a few interesting insights may be posed. 1. Theoretically, terminating only an individual's EC cells while leaving non-EC cells unaffected will result in the termination of the emerged individual, while producing no damage to any system of the host, ergo, death without damage. 2. Further, effectively transplanting an individual's EC to another viable host will result in a successful exchange of an individual's host form. 3. Identifying and isolating the EC will certainly aid in the identification of the hypothesized entanglement molecules. 4. Studying the entanglement molecule could lead to untold developments and technologies. Some creatures on Earth are evolved to terminate even healthy cells once other vital cells undergo necrosis. This is usually done by the release of a chemical death signal that moves through the rest of the healthy portions of the host and causes them to terminate. For creatures that do not possess this self-destruct feature, once the emerged being dies, healthy or undamaged cells of the host may continue to live on. These occurrences suggest that the emerged individual is only linked to its other non-EC cells of the host by a dependency or reliance upon them to maintain vital conditions for continued life. Conditions such as the need for energy and temperature and pressure and vital chemicals that may be required by the systems of the host form for continued function. Therefore, the function of every host for life is singularly dedicated to maintaining the internal and perhaps also the external environment for continued entanglement by the host's EC, thus maintaining the individual's POV. The POV is the composite QEF established and maintained by those same EC. The search for the entanglement cell, or EC, will require the isolation and identification of critical regions of cells that may be referred to as follicle regions. Follicle regions in this context describe isolated diminutive groups of cells which, when sufficiently disrupted, appears to cause the termination of the subject in a manner difficult to distinguish from genuine EC termination. EC, or entanglement cells, being the most fundamental physical implementation of individuality of an emerged composite being, disruption of EC exclusively is hypothesized to result in disentanglement to metamatter, which is deinstantiation, individual death. Follicle regions may actually contain EC, or alternatively, only cells whose function is critical to systemic function, not unlike cells of the heart or liver, only whose role is much less obvious. Determining which of these two possibilities is the case will require the investigation to focus on each follicle group of cells by process of elimination to reduce the group to the barest minimum of effective follicle cells within the group, and then to trace and definitively determine how those remaining follicle cells contribute to host termination. For each follicle group, this process should always lead either to the determination and identification of yet another indirect cause of death or the discovery of the presence of genuine EC within the follicle group. These EC will be those, one or more cells, which contribute only and exclusively to the observed subject termination. This process requires the discounting, not subjected to disruption, of those cells, which either cause intermediate damage to other host systems or do not directly cause host termination. Subject termination due to EC candidate cells within the follicle group must not result in any premortem cellular disruption or non-necrotic, physically or functionally, to any region outside of the follicle group, ergo, death without damage. Approved subjects, flies, nematodes, etc., chosen for this process may need to be high-fidelity clones in order to provide the required consistent physical structure and predictable systemic cellular distribution. This is so the process of elimination may continue unabated with minimal loss of progress as subjects are terminated, and new test subjects are needed to continue the investigation. Further, subjects may not need to be fully formed individuals, but may be sufficiently developed living embryonic forms. These are subjects viable for testing, but not viable by current definitions for independent growth. Probing for the entanglement cell, or EC, does not require physical contact with candidate cells. To the contrary, the astute investigator will quickly realize that the less physically disruptive the probing mechanism, the more progress will result from the exercise. Since the task at hand is not to disrupt any internal cellular function which could kill the cell, but rather to disrupt only the heterodyning mechanism by which the EC maintain the emerged individual POV. The means of disrupting EC heterodyning are potentially numerous, as the monogamy of this delicate state are unforgiving. Infiltration or only identification of the entangled state may occur by the use of appropriate entanglement witnesses, such as properly tailored photonic, electronic, or other non-physical mechanisms. Of course, there is a chance that every cell is an EC. This would require a slight modification of the predictions of the theory, as in such a case the heterodyne state would be far more robust than currently predicted. This is because the entangled state of emerged POV would need to survive the massive changes in cell participation, as cells of the holistic host are perpetually transient. Depending on the relative orientation and positioning of EC relative to other EC, the probe will need to target individual candidate cells or very diminutive groups of the same. This is because it is possible that EC may have developed in close proximity or even in direct contact with each other during the gestation period of initial conception and engaged their heterodyning of their individual QEF to establish the emerged QEF, and then later physically drift apart as the billions of new non-EC cells develop as the subject grows. Alternatively, the heterodyned EC may, in all or some species, remain in direct physical contact with other EC to maintain the heterodyning function required for emerged individuality to persist. Therefore, the probe may need to be focused down to within the diameter of a single cell, and be as non-invasive as possible, yet highly maneuverable as to scan many cellular diameters in rapid succession. Given all of these requirements, the inventive investigator may imagine a probe not dissimilar from the polarized blue or UV laser found in a Blu-ray disc player, and research labs around the world as a good foundation upon which to fashion the probe for this endeavor. The line hypothesis suggests that sufficient disruption of the heterodyne state of EC will deinstantiate the emerged individual, even while the non-EC or even the actual EC remain instantiated, alive as individual functional cells. With all cells of the host remaining fully functional, how is the deinstantiation of the emerged individual determined? There is expected to be a time lapse between POV termination and the first signs of the shutting down of cellular function associated with postmortem necrosis of the host body. The more immediate symptom of deinstantiation will be an alteration in species or subject-specific nervous system and brain functions. Each of such symptoms may be used separately or together to identify POV termination of the subject.